Hello, everyone, and thank you for taking the time to listen into our presentation. My name is Rebecca Halleck. I'm a senior majoring in biology and healthcare administration, and I'm calling from Enfield, Connecticut. I'm accompanied by AJ Lee in North Kingston, Rhode Island, who is a junior and a biology major, and also Dr. Shally Page, Associate Professor of Chemistry in Peterborough, New Hampshire. He will be spending some time in this presentation talking about some of the other students who could not be here today. This presentation is called Hunting an Immune System Gene. In Professor Page's lab, we have been working to discover the functions of a gene called BER2. We will be discussing our research throughout the past 2 years and focusing more specifically on the 2019-2020 semester research outcomes. Next slide, please. The gene we are hunting is involved in the innate immune system. The innate immune system is an ancient system that evolved to protect our bodies from germs and infections. For example, our tears contain compounds that kill bacteria. This investigation centered on a gene of an unknown function, BER2, found in nematode worms that is similar to the immune system genes in humans. Next slide. Throughout our presentation, we modeled regulation, tracked worm survival without the BER2 gene, tested the immune system functions of the BER2 gene by feeding the worms without this gene pathogenic bacteria, and utilized bioinformatics to examine the effects on every gene when the BER2 gene is not functioning. Next slide. In our lab, we use the model organism nematodes, referred to as C. elegans. These worms are microscopic and transparent. They are grown on petri dishes and fed bacteria. Their life cycles include stages referred to as eggs, L1, L2, L3, and L4. L1 are infant worm stages, and L4s are the adults. Their genome is fully annotated. They are cost efficient. They have a short lifespan, fast reproduction rate, and can be frozen, making them easy to grow and maintain. Next slide. Moreover, our research involved BER2, upstream regulation, gene knockdown, gene expression, survival assays, and bioinformatics. Next slide. Throughout our presentation, we will be using some key terms. The N2 worm is the wild-type worm utilized as control in many of our experiments. The wild-type worms eat wild-type bacteria, or OP50 bacteria. RNAi, or gene knockdown, is utilized in our labs to inhibit the gene expression of the BER2 gene in the worms. Therefore, the knockdown worms are referred to as BER2 worms. The BER2 worms eat the knockdown BER2 bacteria. The pathogenic bacteria I mentioned previously is E. faecalis, which is a digestive pathogen. Thus, the survival test was conducted utilizing this pathogenic bacteria. Finally, bioinformatics is a field where computer-based skills are used to analyze DNA and RNA. Next slide, please. We will begin talking about the BER2 gene and its relevance to human genes. This diagram shows 8 human genes. You can see the brown boxes they all have in common. Those are parts of the protein called BER repeats. Next slide. This human gene family has 8 known members. Some are involved in cell division. Some play critical roles in responding to immune system challenges. Here is a cell that is damaged by a pathogen and headed for self-destruction, or a process called apoptosis. This happens if the cell is overwhelmed by pathogen or if the cell is growing out of control. Next slide. Some of the BER family members inhibit this process, even if we want the cell to self-destruct, such as in cancers. These BER proteins inhibit the self-destruction, allowing cancer cells to continue to grow. For this reason, these BER genes have been explored as a way to fight some cancers. So drugs that block the BER protein would encourage the cell to self-destruct. Next slide, please. BER2 repeat regions are similar to the BER proteins in humans and yeast. The one that it resembles most closely is called survivin, a gene that allows cancer cells to survive even when they are cancerous and should self-destruct. Next slide.
So the next section we're going to talk about was work done by Cassie Morgan. We were looking to use bioinformatic tools to look at the sequences near our gene of interest, BER2. And so Cassie Morgan took on that challenge. Next slide, please. So we have here a blue box representing the BER2 gene, and then a red box that represents some regulatory regions that are upstream from that gene. Next, please. So proteins bind to that regulatory region. They affect the regulation of the BER2 gene. And by analyzing the sequences of those regions, the red box essentially, the A, T, Gs, and Cs in that region, we can get an idea of predicting what sort of proteins bind there and if it gives us evidence for our hypothesis about the function of the BER2 gene. Next slide, please. So this is a picture of the results that Cassie came up with from her bioinformatic analyses. This first line is the gene of interest in the nematode, BER2. Those next four horizontal lines represent the genes in the human that are in that same gene family. The last one is a related gene that's actually found in fruit fly. So what Cassie's done is checked for several of these sequences for regulatory regions, and you can see there's 3 here in these colored boxes. And the blue and the green ones are in common with some of these family members, and those are proteins abbreviated as MYC and MYF, and they are involved in immune systems cancer. So this gives us some evidence that they might perform a similar function in C. elegans as they do in these other family members. Next slide, please.
So RNAi, or knockdown, is a process that utilizes to inhibit gene expression of the BER2 gene. RNAi can lead to the shutdown of the gene translation, which allows us to manipulate gene expression. Next slide, please. This diagram shows how the knockdown process works. The mRNA is produced by the cell, and the siRNA is inserted into the cell to affect protein synthesis and ultimately gene expression, as you can see in the blue circle in the middle. Next slide, please.
So this is some work done by Morgan Candon and Sam Casey. This is a survival curve looking at the C. elegans worms. On the x-axis is days of their lifespan, and on the y-axis is the number of worms in the petri dish. The blue line is our wild-type condition worm, but it has the BER2 knockdown in it. And then the orange line is the BER2 knockdown in the presence of the E. faecalis pathogenic bacteria. And what you can see is that they're still increasing numbers. They're still reproducing with the BER2 knockdown, but without that BER2 gene, their ability to survive the pathogenic bacteria is impaired. Next slide, please. This is a representation of some experiments done by Sarah Diltz, who's actually an alumna now. She's gone on to med school, and this is a survival assay over 2 generations of a similar sort of experiment. The blue line is the wild-type bacteria. The orange line is with the BER2 knockdown. The gray line is with the pathogenic bacteria with wild-type worms, and the yellow line is the pathogenic bacteria with the BER2 knockdown. You can see that the survival is impaired, again, in that BER2 knockdown with the pathogenic bacteria. After day 7, you can see a second generation of the wild-type worms increasing and decreasing in population, but the ones that have the BER2 knockdown and the E. faecalis do not recover, so those numbers continue going down. So again, this gives us some indication that BER2 plays an important role in the immune system. Next slide, please.
So an experiment that I performed in the lab began with wild-type C. elegans, or N2s, that were grown on multiple plates, some with the control bacteria, which was OP50, and others with the knockdown, which was BER2 bacteria. Then a 96 well plate was assembled containing variations of the bacteria and worms grown. Observations were taken daily, as well as shaking of the well plate and overnight incubation. Next slide, please. So this is an image of the micro well plate to the right of the screen. So the plate was set up for this experiment involving N2 worms that were initially grown in OP50 or BER2 bacteria, and then each type of N2 was tested in a row that contained either OP50 bacteria, BER2 knockdown, or E. faecalis bacteria. Next slide, please. And this is an image that you can see of one of the wells from a micro well plate that contained BER2 N2s within the BER2 bacteria. Next slide, please. The control worms contained OP50N2s within OP50 bacteria, which showed a high population and mainly normal swimming movements. However, the OP50N2s within rows that contained BIRD2 or E. faecalis bacteria showed decreasing and struggling reproduction rates and irregular movements, such as drastic speed changes and twitching. Next slide, please. BIRD2N2s within OP50, BIRD2, and E. faecalis bacteria all showed struggling populations and irregular movements. By the end of day 16, most worms experienced twitching or very slow swimming. Mainly younger stages of N2s were present as well. Next slide, please. Can you please play the video? So here's the OP50N2s and OP50 bacteria, and you can see their population's doing well with pretty regular movements, just normal swimming going on. This is the OP50N2s within the BIRD2 bacteria, and they're starting to decrease in population, and twitching can be seen in some of the worms. Here's the OP50N2s within E. faecalis. You can see population has decreased drastically, and there's pretty extreme twitching going on within the few worms present. Here's the BIRD2N2s within OP50 bacteria. They're doing pretty well surviving, but you can start to see irregular movements within the worms. Here's BIRD2N2s within BIRD2 bacteria, and you can see a lot of presence of dead worms, which are the straight worms that aren't moving, as well as the worms starting to have irregular movement. Here's the BIRD2N2s within E. faecalis. There's a large decrease in population and barely any later staged worms. It's mainly the L1s and L2s.
Next slide, please. So at this point, we've got some pretty good evidence that this BIR2 gene is playing a role in the immune system. It's helping to, in some way, protect the worms against this pathogenic bacteria. So we developed a hypothesis that it might be involved in this TLR4 pathway for the immune system. In this picture, we've got TLR4 receptors that are in pink crossing the membrane, and when bacterial signals, represented here by the LPS, click please. Next slide.
So lipopolysaccharides.
Go ahead, AJ.
All right. So lipopolysaccharides, or LPS, were added to the well plate. Two forms of LPS were added. LPS-RS, which is an antagonist that inhibits the TLR4 pathway, and LPS-EB, which is an agonist that stimulates TLR4 pathways. These were added to all wells, with the exception of the LPS-EB within the E. faecalis wells. Next slide, please. When the LPS-RS was added to both E. faecalis wells, that resulted in increasing populations and movements becoming more normal. LPS-EB was also tested, but not enough times for significant data to be recovered, but they did display lower survival rates. Next slide, please. This graph displays the effects of LPS-RS that had on the N2 population in E. faecalis wells. BIRD2N2 wells that had E. faecalis and LPS-RS showed that the population more than doubled, while the OP50N2 wells that had the EF and the LPS-RS showed little change in population. However, the OP50N2 wells that had E. faecalis without the LPS-RS had almost no worms present, with the exception of one well that showed high population, which caused our data to be altered. The population increased with LPS-RS. The population increase with LPS-RS is consistent with our hypothesis that we are blocking that pathway. Next slide, please.
So spring break arrived, and AJ set up this experiment, and I continued working on it, which is using not only the agonist, but also the antagonist for this pathway. This is a slide at the next plate showing that the survival is worse with the BIR2 knockout, as we've seen before, but significantly worse with the pathogen and the BIR2 knockout. So significant difference there. Next slide, please. the experiment with the LPS-RS and the LPS-EB, which is the agonist that should trigger this pathway, we saw a trend that would support our hypothesis, but the results were not statistically significant. So this is an experiment that we would need to repeat when we manage to get back to campus in our wet lab. Next slide, please.
So overall, the efacalis was most detrimental to the population of the BUR2 and OP50 N2s grown. BUR2 bacteria also inhibited the populations, but to a less extreme as the efacalis. Both bacteria caused the N2s to move irregularly, and the presence of the LPS-RS resulted in increasing population and normalization of N2 movement, regardless of the N2 or bacteria, regardless of the type of N2 or bacteria present. Next slide, please.
So this is an experiment that was conducted by Morgan Candon. This is looking at some other types of pathogenic bacteria. She was looking at a Salmonella typhimurium. Some of you might be familiar with Salmonella as a toxin in foods, and these bacteria grow. And she was working to reduce experimental error by using just the adult synchronized worms. Next slide, please. So this is a graph of those results, looking at, in the blue line is our pathogenic efacalis bacteria with the wild type. The orange line is the Salmonella with the wild type worm, and then the gray line is the efacalis BUR2, and the yellow line is the S. typhimurium BUR2. And so we see a little bit of a trend there, but no significant differences at this point with the Salmonella. So we stopped using that bacteria, and Morgan moved on to some alternative worm strains. Next slide, please. So here we see a representation in a blue circle here of an AKT gene, which is a protein which is important in longevity and stress response. The gray box in the middle, TRF1, is a gene that's involved in the immune response. And the green box in the third one is upstream of one called SED3, which is involved in cell death, apoptosis. And so by looking at these alternative worm strains, we're hoping to be able to distinguish what's really going on with the BUR2, whether it's involved in the stress response, whether it's involved in the immune response, or whether it's involved in apoptosis or some combination thereof. Next slide, please. So this is Sam's work, and Sam showed that in all of these conditions with the TRF worms, that their survival was impaired. We didn't see a significant difference between the survival of those with and without the BUR2. So we stopped using those TRF1 worms, and we're still looking for a good model system where we can distinguish between those pathways. Next slide, please. So one way that we can actually measure how much of these genes are being expressed is a technique called RT-qPCR. So this is a method of really measuring how much mRNA is produced, so how active that gene is. And the way that this works is that during the cycle, there's a fluorescent signal that's there in the sample, and as it's amplified in the instrument, it becomes double-stranded and the fluorescent signal binds. So this allows us to have a fluorescent signal that's proportional to the amount of RNA present. Next slide, please. So this is showing a typical sort of response curve from this qPCR instrument. They start off at 0 fluorescence, and as the RNA gets amplified, at a certain point, the fluorescence goes up, and you can see these red lines going up on the right-hand side. And at a certain point, they pass the threshold, which is the upper horizontal line, and that's the point where we measure it. So the sooner they cross the threshold line, that means that they took fewer cycles to amplify, which means that there was more RNA there. So this is a way of quantitating how much gene expression there really is. Next slide, please. So Sam did an experiment where she was looking at OP50 knockdowns of BUR2 as well as family member BUR1, and looking at expression of SED3, which is the apoptosis gene we talked about, and comparing it to our control, which is the gene expression of something called actin, which is a housekeeping gene. Next slide, please. So you can see each of those wells has its own little amplification curve. So she has a whole bunch of samples here. And in the bottom graph, you can see that the BRCA2 gene expression is lower, which is simply a nice confirmation that these experiments that we're doing, knocking down BRCA2 really is knocking down that gene. And then the graph on the right shows looking at those expression levels in the presence of the BRCA1 knockdown and the BRCA2 knockdown. And Sam did not see any significant differences with the BRCA1 knockdown, and maybe something approaching significance with the BRCA2 knockdown, where it might be upregulating that gene involved in apoptosis. And again, probably worth, this is preliminary data, probably worth replicating this again. Next slide, please.
So my research this semester utilized bioinformatics tools to analyze the RNA-seq data and measure gene expression similar to qPCR so that those techniques could be compared. Next slide, please. So bioinformatics is a field of science that uses computer technology to analyze complex biological data such as genetic code, protein sequencing, proteins, and protein structure. Here we are using these computers to analyze amounts of RNA in our samples. Next slide, please. So my research experiment utilized bioinformatics to analyze the mRNA from the worms. mRNA is a relevant indicator of gene expression, which can be explained using the central dogma theory of translation and transcription. This image describes the central dogma theory. It is important to my experiment because of the concept of reverse transcription. A sequencing technique referred to as qPCR was used to reverse transcribe the mRNA and match these fragments to the annotated worm genome. Next slide, please. Bioinformatic techniques allows us to count the number of RNA fragments, also referred to as reads, and ultimately measure gene expression. Next slide, please. So this diagram depicts my research setup. Sample 1 were worms that were fed wild-type bacteria and transferred to plates with wild-type bacteria. Sample 2 worms were fed knockdown bacteria and transferred to plates with knockdown bacteria. Sample 3 worms were fed knockdown bacteria and transferred to pathogenic bacteria. And finally, sample 4 worms were fed pathogenic bacteria and transferred to pathogenic bacteria. Each transfer was done with a single generation of worms using L1 filtering. Next slide, please. The purpose of my experiment was to use bioinformatics to get a more specific measurement of gene expression. The overall goal was to analyze gene expression, and the more copies of mRNA of the gene indicated the amount of protein production, thus measuring the magnitude of the expression of the gene. Next slide, please. So these steps listed above were involved in setting the controls for the experiment to ensure only one generation of worms and an equal number of worms were used for the sample collection. Next slide, please. After transferring the worms were collected and their RNA was purified. These RNA samples were diluted to 100 nanograms per microliter and then sent to UNH for the RNA sequencing technique. Next slide, please. The RNA sequencing process begins by adding adapters to the ends of the RNA samples. These ends are attached to a plate. Fluorescent nucleotides are added to that plate, and a picture is taken above after each nucleotide is added to the plate. The nucleotide sequence is recorded, and the data was put on a server for us to access. Next slide, please. So this diagram summarizes the process I just described. As you can see, the RNA was isolated from our samples, prepared for sequencing. Sequencing was then performed, and you can see the little plate in that black box next to the big computer, which shows the different colors of nucleotides. The data files were collected and logged as FASTQ files, and those were the end result that we got from this testing. From here, bioinformatics was used to evaluate the data. Next slide, please. So as we previously described, my research is the beginning of or is being used to compare our qPCR measurements and gene expression. Next slide, please. So this is an example of the number of reads I received back from UNH. So there was 25 million reads. They were all about 150 to 200 nucleotides long, making us having access to 37 trillion nucleotides of data from my samples. Next slide, please. So overall, my bioinformatics workflow was to obtain the data files, do a process called FastQC, use Termematic, HiSat2, StringTie, BallGown, RStudio, and finally have an end result of data graphs that we can analyze. And I'm going to get into each one of those programs. Next slide, please. So FastQC is used for quality control. If you look at this top graph on the right, you'll see that these are the lengths of reads, and the majority of it in the first half is in the green section, which is what you want, and then you see this big spike that goes into the yellow and red sections, indicating poor quality of reads. This top one was before we used Termematic, and this bottom one was after we used Termematic, which trimmed the poor quality reads. And you can see that the majority of or actually all of the read now is in the green good quality section. Next slide, please. So this was Termematic. It trims the poor quality sequences and the adapters. If you look at the very last thing, in this picture, it shows that the program is running. Next slide, please. So HiSat2 pairs and maps the sequences to the genomes. If you look at this very last number on the bottom, it says 96.52% of overall alignment, which is very good. We want to try to match the reads as much as possible to the genome. Next slide, please. So SAMtools was used to sort and prepare the HiSat2 results for the StringTie program. And you can see it was merging files at the very bottom, and that's the command for it listed above. Next slide, please. So StringTie counts the number of replicates of a specific gene. We are currently at this step. We have reached a couple of bumps along the way and just trying to figure out the programs and how to make them work the way we want them to. But our next step would be BallGown, which prepares the data for visualization of RStudio. Next slide, please. RStudio allows us to create visual representations of the data and easily analyze the data. Next slide, please. So, as I mentioned, we are currently in progress with our bioinformatic results. So far, we have good FastQC results after Termematic, indicating Termematic successfully trimmed poor quality reads. And we have overall good alignment from HiSat2, as well as an indication of pure C. elegans RNA samples, which means that we don't have any contaminations in our samples, which is what we want. Next slide, please. So from my experiment, we expect to be able to produce volcano plots, which show us what genes are affected by BIR2 knockdown and what genes are affected by the presence of the pathogenic bacteria. In the volcano plot here, which is an example, we see the p-value on the y-axis and the gene concentration on the x-axis. The blue dots are genes that were statistically significant and downregulated, and the red dots were genes that were significantly upregulated. The plot produces a gap in the middle because the gene expression of these genes was not statistically significant. From our volcano plots, we will be able to compare our results to SAM's qPCR data. And finally, we expect to make a conclusion about BIR2 gene expression. Next slide, please.
So in our next steps, we have a lot of work to do still. We need to get some more qPCR data and get some better precision with those data, smaller error bars. We need to repeat our LPS survival experiments. We also are acquiring some equipment to do some analysis with the C. elegans apoptosis worms. This picture here is of a worm that when its cells undergo apoptosis, actually glows a nice green. And we can use the Dartmouth fluorescence microscope to document that. We want to finish analyzing our gene expression data and next year move on to some protein measurements. In addition, I have someone starting the summer who's going to be looking at other immune system genes of unknown function to analyze. Next slide, please. So want to provide some acknowledgments for all this support and work. Most of the funding for this work has been provided by New Hampshire INBRI, and the qPCR machine that we used in particular was provided by Stephen Firing, who's a New Hampshire INBRI administrator at Dartmouth. At Franklin Pierce, Lisa Bomba provides technical support, and Professors Koenig and Patty Arsenal provide administrative support for us in the science department. For our bioinformatics, we've received excellent technical and bioinformatic advice from Devin Thomas and Joseph Savigny at UNH, Benjamin King at University of Maine, and Joel Graber at the Jackson Laboratory. Next slide, please. And this is, of course, the researchers that we've been talking about in my lab. Up in the top left there is AJ, working hard at the scope. And then on the upper right is Rebecca with her poster last year. The bottom left is Cassie, who did the bioinformatics for the regulatory regions. And bottom right is Sam and Morgan, who did some of the survival assays that we talked about. They all did a great job, worked hard. Next slide, please. Thank you for your attention. It's a pretty narrow little area of investigation, but we're working hard and we're really interested in seeing how these results will translate to human immune system and cancer genes. So thank you again.